Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in comment below. So today we will discuss about the crassulacean acid metabolism in the plant. So first we will discuss about the intro of it. So carbon dioxide fixation in succulent plant, how it's possible. So that's the crassulacean family, which that's contain a cactaceae family, orchidaceae, uh, bromelaceae, liliaceae and euphorbiaceae, which they are family related to the nighttime stomata open and daytime stomata closed due to prevent the loss of water because they are present in the arid atmosphere. So that is mean that's arid condition which less required water because not water too much desert ecosystem. So the sunlight will be fall on the 90-degree angle into the earth in the equator of the earth. So that's why it's important to know about that. The light will fall more, so a plant will prevent to loss of water because the water requirement is the unavailable as much as like another uh, climate plant, for example tropical subtropical region. So their leaves thickness and leaf surface area is low as compared to the high surface area in the semi-arid. So arid will contain less. So here I draw a earth which contain arctic a region, a polar and subpolar region, and tropical region and subtropical as well as equator. So for example near to the equator, so the 90-degree angle light fall on the 90-degree angle and here, so that's why the light is too much. It means the prevention of stomata is necessary for prevention of loss of water. So important to know that is the CAM plant opens stomata on night time and fix carbon dioxide into organic acid during night time. So remember it. These help to minimize the water loss due to transpiration. So transpiration is like a harmful for it for this plant. So their leaf structure is different as compared to the C4 plants, C3 plants. So this is also known as the C4 plant because the malic acid which that is contain carbon 4. So the 4 carbon containing acid will be formed. So important to know that here is I will draw a cross section area of the leaf which that is understand as well. So the mesophyll cell and bundle sheath cell is similar as much as like a mesophyll. So the spongy and palisade mesophyll cell is majorly equal to each other. So here is the cross section of the leaf contain epidermis, upper epidermis. On the other hand, the lower epidermis is downside, and that's here is the vein which contains xylem and phloem into the leaf. So here is the mesophyll cell. On the other hand, the bundle sheath cell quite similar to the mesophyll cell. So the mesophyll cell is similar to the bundle sheath cell. So bundle sheath cell is wrapping around the vascular bundle, for example phloem and xylem. So here is the air space are present and here is the stomata which that is contained by the guard cell. It can be 4 guard cell or 2 guard cell, pair of guard cell. So here I draw a structure of cell which that is used for the understanding the cycle of C4 or CAM plant. So the CAM plant contain here is the chloroplast, mitochondria, nucleus and vacuole and cytoplasm in it. Into the and outer membrane cell membrane surrounded by the cell wall. So the cell wall is made up of polysaccharide of cellulose. So the sugar alpha to beta subunit of sugar will be formed, beta sugar and alpha sugar. So here I will draw a chloroplast is here and here is the mitochondria, and on the other hand here is the vacuole which that is and that is are present in the cytoplasm, and here mitochondria and chloroplast, and now I will draw a vacuole. So for understanding easy to understand with this diagram. And you can say here is the chloroplast and mitochondria and vacuole. So here is the starch are present into the leaf or into the plant, which convert into the sugar during daytime. So that's mean daytime will sugar conversion. A starch are making photosynthesis produce sugar. So that's malic acid will be formed during night time by the conversion of sugar from the pyruvate. So that is the feel like a bitter or acidic feeling of their leaves. So here is the person which that is checking the taste of leaf, which that is mean the sour. So on the other hand, now we will discuss about the light reaction during the daytime in the thylakoid, into the thylakoid membrane. So why it is why it, because it is made up of chlorophyll. So chlorophyll absorb light and water hydrolysis to produce NADPH ATP, and these NADPH and ATP used into the Calvin cycle to produce by the fixation of carbon dioxide to produce starch or glucose. So this mechanism is same as much as equal to the basically C3 plant. But here we will discuss about the CAM plant. So whole process we will understand, and next we will able to distinguish these differences. We can differentiate. So here the starch which that's convert into the glucose is known as the um, glucogenesis. So the glucose will be formed from the starch. On the other hand, that is here is the myeloplast is contains starch synthetase enzyme to use starch synthesis. So this is the myeloplast is the storage organelle in the plant, which that is also found in the tubers or some at roots, etc., or seeds. So here is the glucose also converted by the glycolysis to triose phosphate. It means the triose phosphate, triacylglyceraldehyde phosphate. So on the other hand, glucose also converted by the glycolysis to produce pyruvate. So these pyruvate will move into the mitochondria. So then when move into the mitochondria to convert acetyl-CoA, and this acetyl-CoA will go enter into the basically citric acid cycle by the fixation of oxaloacetate, which that's convert oxaloacetate by the 2 carbon more fixation from the acetyl CoA to produce ATP and FADH2 and NADPH2. This is the useful for the electron transport chain to produce more energy. So on the other hand, carbon dioxide we release into the mitochondria, or hydrogen bicarbonate is also release carbon dioxide into the cytoplasm to go uptake into the chloroplast. And here is the stroma which contain a Calvin cycle. Calvin cycle means Rubisco enzyme are present which that fix the carbon dioxide. 3 molecule of carbon dioxide obviously fix to produce sugar, and this sugar is the starch synthase enzyme to produce uh, starch. Here is the light reaction and dark reaction which occur. So dark reaction produce again starch to convert into the pyruvate by the glycolysis when did that. So that's important, and that is the pyruvate will also move into the outside of the outside of the chloroplast. So this all thing is during the C3 plants, which that is the chloroplast. So on the other hand, here is the basically phosphoenol pyruvate, which that is fixed by the carbon dioxide to use ATP to produce oxaloacetate. Now we are discussing about the CAM plant. So here is oxaloacetate is produced a 4 carbon containing compound, and this malate dehydrogenase enzyme used to it by the dehydrogenation of NADPH will be released into make a NADH. So this NADPH will be formed And malate dehydrogenase enzyme used to convert the malate uh, ions, so malate compound. So in this way, here is the NADP plus will convert into the NADPH. So here is the wrong. Basically here NADPH will be generate, not will be used. So remember it. So that is the light reaction into the into the presence of, basically if we will talk about the NADPH will be produced from the chlorophyll light reaction is also. So that is important, and malate ion will move inside into the vacuole, and that vacuole will be accept this malate ion, and two higher proton will be enter into two proton will move inside, and one require to one malate move inside to produce malic acid, and this malic acid is the 4 carbon containing compound during night time, and that stored into the vacuole is just storing, and that is night time. After the daytime, so nighttime is the acidic medium will be create. After the night time will be daytime begin, and daytime will release the malate by the um, two proton release separation of two proton. So the one potassium ion move inside to back move of the two proton outside of the vacuole into the vacuole from the vacuole. So here is the malate which that is produced, and this malate go back into the during the daytime into the chloroplast. So that's again will be produced pyruvate by the pyruvate dehydrogenase enzyme to produce NADPH from the NADP plus. So it will produce pyruvate, and this pyruvate will again go into the mitochondria, either go into the mitochondria or again gluconeogenesis process to make glucose, and that starch synthase enzyme used to produce a starch. So the starch is the storage of the storage molecule in the form of glucose are present in the plant. So that's all thing is done, and here's the triose phosphate will be produced again, or pyruvate produced, and that's go into in chloroplast to out to the cytoplasm in the form of phosphoenol pyruvate, and this phosphoenol pyruvate will move here. So this means the malic acid stored into the vacuole during night time is the 4 carbon containing compound. So that's important. So thanks for watching.